Most biotech startups burn their first 2 years and then their first $2 million figuring out things that a drug discovery CRO already does every single day. The chemistry decisions, the ADMET screens, the process route, the analytical package, and by the time the startup realizes they need outside expertise, they are already 18 months behind their original IND target. Today, you are going to see exactly what happens inside a drug discovery CRO, the 5 core steps, what the science looks like, and where early stage teams most often hit the wall. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with biotech founders and early stage pharma teams every day to move drug candidates from initial chemistry through IND ready packages. Today, here one of our drug discovery CRO series, we are pulling back the curtain on the whole process. Here is the uncomfortable truth about the drug discovery. 90% of the drug candidates fail, and most of those failures are seeded in the earliest decisions. Not at the clinic, in the lab at month 2, when a synthetic route chosen for speed rather than scalability, or when an ADMET flag is noted but not acted on. Small biotech teams try to build these capabilities in-house, and the gap between what they need and what they have compounds quickly. A CRO partnership exists precisely to fill that gap, bringing in the expertise, the platforms, the throughput that would take years and tens of millions of dollars to build internally. 5 core steps. So what does a drug discovery CRO actually do? There are 5 core steps, and understanding all 5, not just the synthesis piece, is what separates the startup that hit their IND timelines from the ones that don't. Step 1 is target and hit identification. Finding the candidate molecule that interact with your biological target. Step 2 is hit to lead chemistry. Iterating on structure-activity relationships to find the lead series. Step 3 is lead optimization, shaping that lead for the clinic, potency, selectivity, ADMET. Step 4 is process chemistry, converting your discovery route into something scalable and GMP ready. And step 5 is analytical characterization, the complete scientific package that backs every candidate you advance. Hit identification is where a lot of early stage teams underestimate the depth of expertise required. It's not just about having access to compound library, it's about knowing which computational filters to apply, which fragments to prioritize for synthesis, and how to read the first binding data intelligently. Good CROs bring docking and pharmacophore modeling tools, proprietary fragment libraries, and increasingly AI-assisted trials that cuts the number of compounds you need to make before finding a genuine lead. Then in hit to lead, the real medicinal chemistry starts. Weekly synthesis and testing cycles, each round teaching you something new about what the target wants from the molecule. Lead optimization. Lead optimization is the hardest mile in the drug discovery, and it's the one where the most time and money disappears. You have found a molecule that works in a biochemical assay. Now you have to make it work in a body. That means turning potency into a nanomolar or even picomolar range while simultaneously checking that it doesn't hit 200 other kinases and that it survives the liver for long enough to reach the target, that it crosses the relevant membrane, and then that its structural features don't predict HERG liability or genotoxicity. A well-equipped CRO runs all of this in parallel, not sequentially, which is what compresses the timelines. The teams that run them sequentially usually find out about their metabolic stability problems 6 months after they should have. Process chemistry, which brings us to the process chemistry, the step that discovery-focused teams most consistently underinvest in until it's too late. Discovery routes are designed for speed. At milligram scale, they use whatever reagents get the molecule made fastest, even if those reagents are expensive, hazardous, or would terrify a GMP manufacturing team. Process chemistry re-engineers the synthesis from the ground up, optimizing yield, replacing difficult intermediates, eliminating safety hazards, and establishing a route that can be reproduced at gram, then kilogram scale. Lock that route before GMP manufacturing. Any change after GMP entry triggers a validation exercise that adds months to your timeline and cost that no early stage budget expects. Now, where startups hit the wall, the failure patterns are consistent enough that they have become recognizable. A hit to lead stall with no all in-house medicinal chemist, a problem that drags for 18 months until a CRO engagement compresses into 4, and admit failure at IND filing because metabolic stability was flagged at lead stage and then deprioritized, leading to a full reformulation effort that nobody planned for. A synthesis bottleneck because the discovery route was never assessed for the process transferability and the GMP CMO simply cannot reproduce it. At Resolve Mass, we have rebuilt discovery synthesis routes for the process scale-up in under 8 weeks, specifically to unlock IND timelines that had stalled exactly like this. The cases look different on the surface, the root cause is almost always the same, the decision deferred in discovery that become a crisis closer to the clinic. Analytical coverage. Step number 5 is analytical characterization. Closes the loop on every candidate you advance, and it's not optional. A molecule without complete characterization is not a drug candidate, it's a liability. NMR confirms your structure, high resolution mass spectrometry confirms molecular formula, HPLC purity needs to be above 95% for anything entering a biological assay. If your CRO is handling you candidates at 85% purity, that's a conversation you need to have. Chiral analysis matters from the moment your molecule has a stereo center, and every batch needs a proper certificate of analysis and batch record, not a handwritten note. The integration piece is what distinguishes a drug discovery CRO from a contract synthesis shop. The data from analytical feeds back into lead optimization decisions. The process chemistry inputs from the analytical development, and the full package synthesis, characterization, ADMET stability is what your regulatory team will lean on when they write the IND. So what should you walk away with the discussion today? The decision made in the first weeks of discovery echo all the way to IND, often 18 months later. A CRO accelerates all 5 steps, not just synthesis. The value is in the integrated expertise. Admit profiling at the lead stage prevents the expensive surprises that show up at the IND filing, and process chemistry must be designed before you approach a GMP manufacturer, not discovered during the first engineering batch. Next videos, we are going to get specific on medicinal chemistry for startups, how to design and run assay cycles when you don't have a full-time medicinal chemist on staff, and what to ask your CRO to track on your behalf. If you are an early-stage biotech or pharma team and you need a drug discovery CRO partner who can take you from target hit through IND-ready synthesis and analytical packages, Resolve Mass Laboratories work with small and mid-sized programs across North America, from first hit identification all the way through process chemistry and GMP handoff. Thank you so much for watching this session until now. Goodbye for now and see you in the next one.